everyone, my name is Hannah Davidson, and this is my presentation for the Genetics for Health Professions course. Today I'm going to be talking about hereditary hearing loss type DFNB1. So as a quick overview, individuals affected by hearing loss have been documented since the BC era, so this is not necessarily a new condition. However, at present we have a lot of different ways for characterizing hearing loss. Hearing loss can be characterized by whether it's syndromic, meaning it happens in conjunction with a lot of other bodily symptoms not explicitly related to hearing loss, or non-syndromic, meaning that it occurs in isolation on its own. It can be characterized by its physiological etiology, so that means like the underlying physiological basis for why the hearing loss occurs. That can be conductive, meaning that it's affected by structural changes to the external part of the ear. It can be sensory neural, which refers to structural changes to the part of the inner ear. It can be mixed, which is a combination of those two types of etiologies. And then there's also a condition called auditory neuropathy spectrum disorder that I won't talk about today. It can also be categorized by the extent to hearing loss, so people can have mild, moderate, or severe hearing loss depending on decibel threshold. And then you can also have hearing loss that is congenital, meaning that it was present at birth, or acquired. And then, because this is a genetics class, and I'll be talking mostly about the genetics today, even given all of these different categorizations, at present about 50 to 60% of congenital hearing loss is genetic in nature. So for this project, I'm specifically focusing on non-syndromic hearing loss and deafness, which is also known in the clinical literature as DFNB type 1. This type of hearing loss affects approximately 1 to 3 in 1,000 newborns in the United States, and there's about 90 genes associated with this kind of deafness. However, within that 90, the most common gene mutations that are associated with DFNB1 are the GJB2 gene mutation. That gene is on the 13th chromosome, as well as some of these other ones I list here that I won't get into because I will be focusing on DFNB1 that's caused by JJB2. In terms of treatment and considerations, is that non-syndromic hearing loss is not fatal. And so it's not something that can, affect life expectancy, mortality, anything like that. However, there are documented cases of hearing loss when not properly identified early on can sometimes result in challenges with reading comprehension and learning disabilities. In terms of available treatments, this is where the conversation gets kind of interesting. Cochlear implants were really developed in the late part of the 20th century, so they're relatively new technologies, and they're noted as sort of the first successful treatment of deafness. They essentially use a combination of a transmitter, a microphone and speech processor, and electrodes to essentially assist the ear in processing and transmitting sounds. And so what they produce is not exactly sound in the way that those of us who are, born hearing would understand them, but they produce signal that can register as sound to the brain. However, they in themselves are not without controversy, in part because over time there's evolved this really rich cultural community around deafness. The deaf community, within the deaf community, hearing loss is not necessarily seen as an impairment that needs to be fixed. The deaf community uses generally sign language to communicate with each other, and the emergence of cochlear implants onto the clinical scene has been seen as sort of threatening to the way of life for deaf people. So thinking about the molecular cause of hearing loss, the gap junction protein beta 2 has a length of 681 base pairs. This encodes about 227 amino acids. And specifically, when it's functioning properly, GJB2 encodes connexin 26, which is a gap junction protein that essentially permits the travel of potassium across the cell membrane. Those of you who've taken Anatomy and Physiology 2, or Cell Bio, or even Neuroscience, know that maintaining a homeostatic potassium gradient in the inner ear then is really necessary for sound waves to be effectively processed by the brain. And so this is also a little illustration of what connexin 26 looks like when it is functioning properly. So altered connexin 26 function in DFNB1 is generally caused by point mutations to the JGB2 gene. The most common is nucleotide deletion that happens at position 35 in the genomic sequence. This type of mutation generally informs the sorting of connexin 26. So as a result, connexin 26 loses function entirely because it's not sorted into the correct place. And so other mutations do, as I mentioned, occur with connexin 26. And a lot of these don't necessarily result in as radical of loss in function. Oftentimes they result in slightly altered functionality, which may mean that there's some hearing that's present, but it's still weaker than it would be. And so then in terms of the inheritance pattern, DFNB1 mutations occur via autosomal recessive inheritance. And so I thought for my presentation I would do some kind of fun exercises, sort of talking about inheritance pattern and sort of seeing how this actually plays out in people's real lives. So my first scenario, Joan and Bill don't have any issues with hearing. Their first child, Emily, was born with hearing loss, identified at her newborn screening, which was later found to be related to a mutation in the GJB2 gene. What is the likelihood that their subsequent children will inherit the same kind of hearing loss? So first, I have Joan and Bill's genotypes. We know that they are both carriers because if they themselves are not symptomatic, then they both have to be carriers to be able to have an affected child. This is what the Punnett square looks like. So based on this Punnett square, we know that their next child has a 1 in 4 chance of being a non-symptomatic non-carrier. Their child has a 1 in 2 chance of being a non-symptomatic carrier, and a 1 in 4 chance of being symptomatic. So they'll have hearing loss. And then I have Denise and Rhonda, who are a queer couple, and also members of the deaf community with the GJB2 gene mutation. And so they decided to work with a sperm donor who is deaf and has the same GJB2 mutation to conceive their children. And I'm asking, what is the likelihood that children carried by either Rhonda or Denise will be deaf? And so you have the genotypes here. Rhonda, Denise, and the sperm donor all have the same genotype for the GJB2 mutation. So this is what the Punnett square looks like. This one's a little bit easier than the last one. Their children have a 4 in 4, 100% chance of being symptomatic. So all of the children will have hearing loss or deafness, regardless of whether Rhonda or Denise carries the pregnancy. And so this is what a pedigree of a family might look like in the context of inheritance of DFNB1. And so you have the parents who are both affected, therefore all of their biological children are also affected by DFNB1. So in terms of identifying hearing loss, screening is one of the kind of biggest ways that we approach identifying hearing loss in the United States. However, practices on newborn screening for hearing loss vary by state in terms of whether it's done when someone is immediately born as sort of a part of postpartum follow-up. However, at this point, about 96% of children in the United States do receive some sort of hearing screening before the first month of life. So screening itself and sort of the nationwide implementation of screening does not actually include genetic testing. Instead, we make use of 2 different technologies most frequently. The otoacoustic emissions test, this is what's shown in this picture here with this cute little baby. An auditory brain stem response test is also sometimes used. However, usually the OAE test is the first line of screening, and then an ABR test may be ordered after the fact if there's an abnormal OAE test in order to see if there is a response in your brain to certain hearing stimulus. In terms of identifying hearing loss from a genetic testing perspective, genetic testing allows us to identify the genetic basis for hearing loss. Like I mentioned, we don't tend to use genetic testing a lot in the identification of hearing loss in newborns. Usually a lot of the concerns that are most immediately intervention strategies, how to support those infants and their families with language acquisition, especially if this is a child who is born into a hearing family. And the underlying genetic basis is not always necessary in terms of really getting families connected with those resources. However, some people may want to know the genetic basis for the hearing loss, and this can be especially helpful for families who might be making family planning decisions, so think back to the pedigree that I mentioned. Generally, there are single gene options available that might look particularly for the GJB2 gene, especially because that is the most common mutation. However, multi-panel options are also available and are generally recommended, in part because, like I mentioned earlier in my presentation, there's over 90 genes that are connected to hearing loss, and particularly DFNB1. And so what is sort of understood as like the gold standard in terms of genetic testing for hearing loss is to test for all of them in terms of identifying a genetic basis for hearing loss. However, there's been sort of an emergent discussion of incorporating genetic testing into newborn screening, but this is coming with some controversy, both in terms of whether it is really cost-effective to incorporate genetic testing for hearing loss when, again, we have these relatively affordable tools that have been really helpful in identifying hearing loss and getting families set up with adequate resources. And also there is a very complex history of genetic testing in relation to deafness where, as we've read a little bit about with eugenics in our class, and also, again, with the fact that there are some very strong, really powerful opinions on the side of the deaf community in relation to sort of the clinical pathologization of deafness, that there are some individuals within that community who believe that genetic testing still kind of harkens back a little too much to the eugenics movement. So that is my project. This is my source slide. And then this is my source slide for images, which I did in order of appearance as opposed to enumerating them. All right, thank you all so much.